So I'll move on to the second step of the information transfer in the so-called central dogma of molecular biology. We have already looked to a significant detail about DNA replication, and we will now move on to looking at the processes of transcription and translation. Okay, and the molecule we look at is DNA-dependent RNA polymerase. All right. So let's make sure before we look at transcription about what exactly a gene is and what is the relationship between a gene, DNA, and chromosomes. And obviously, all of you have done biology in 11th and 12th would have a more clearer understanding of this as compared to the students who dropped biology in 11th and 12th. So think of it as a one slide refresher. So this is a nice pictorial view of the relationship between double-stranded helical DNA, which you have seen a lot about, and a chromosome. So a chromosome literally is nothing but a single long strand of double-stranded DNA, which is folded and compacted into a structure like this. Now, it's important for you to understand that in a cell at any time in your body, DNA is not really in this form or shape. Chromosomes are seen, and this high level of packing of DNA into a chromosome is usually a process done just before cell division, simply because a compact chromosome DNA entity, which is basically a chromosome, is easier to move around, especially when you have decided to break your house into two, give half of your material goods to one house and other half of your material goods to another house. So having a chromosome, which is like a packed suitcase, makes it easier to push it around and carry it around. It's as simple as that. So under normal circumstances, in a, let's say, functional differentiated cell in your body, which is not going to undergo mitosis, DNA is actually in a very unpacked and diffuse state. And in this unpacked and diffuse state, it is still very much in contact with the nucleosomes, and it is in the form of a chromatin fiber. And this, of course, is true only in eukaryotes and not in prokaryotes. And when you look at naked DNA, which is what we are doing now, which is not the form in which it exists, there are always DNA binding proteins which are associated with DNA. Then we can take a stretch of DNA, as shown over here in pink, and call it a gene. All right. And in a simplistic and not too wrong visualization, as you look along the length of a double stranded piece of DNA, you will have genes along the length of the DNA. And you can basically give them numbers or names called gene 1, gene 2, gene 3, gene 4. Fine. So here we are dealing with a double helix, which is an antiparallel double helix. You have information coded in one strand, and for redundancy, complementary information coded in the other strand. Each strand is going in a different orientation. Let's just label it as 5 prime, 3 prime, and 5 prime, 3 prime. Fine. Now, I've already told you that there are approximately 3 billion bases in a human genome. And for every animal, plant, bacteria, archaea, viruses, the number of bases differ. Now, we also know that there are approximately 20,000 protein coding genes in the human genome, and approximately, again, let's say 20,000 again, the number is very precise, which is shown over here, non-coding genes. Now, what is the difference? The genes, and this is one gene, will code for an mRNA, which in turn will code for a protein. Okay, so this becomes a protein coding gene. Whereas there are genes which will code for a piece of RNA, but this piece of RNA will not go forward to make a protein. It will remain as RNA, and we call these non-coding genes. Now, very clearly, I have been emphasizing the fact that a stretch of the double-stranded helix is a gene. And since we know approximately that there are 3 billion base pairs in the human genome, and we know how many genes are there, and I'm approximating approximately 30,000 genes, which is an approximation. On average, each gene will occupy 10 raised to 5 base pairs. This should be pairs. This is a typing error. I'm sorry about that. Now, it turns out that genes are hugely variable in length. For example, the longest gene in the human body is a gene for a protein called dystrophin. And this region encompasses 2 into 10 raised to 6 base pairs. So that's a huge amount of a length of a gene, basically 2 into 10 raised to 6. At the same time, you will also have small genes, let's say 10 raised to 2, which are 100 base pairs long, not too long at all. So you have a wide range of genes, and a gene, the way we are defining it over here, is basically a stretch of a double stranded helix, which codes for RNA. And in some cases, like this, the RNA continues to code for protein. And in some cases, like this, the RNA does not continue to code for a protein. So this is straightforward and simple enough. Now, let's look at the structure of a eukaryotic gene. And this is again simplistic, but it's very accurate. So now this is a stretch of DNA, double stranded DNA. And we basically will look at one strand of the DNA. And we'll worry about which strand we are looking at, because each strand has complementary information. And we usually read off one strand, and the information makes sense on one strand. Now, in this stretch, we will basically say and start giving nomenclature and defining regions. Okay. So each gene, and now this is very approximate, this is a number I have created, which is, let's say, 10 kilobase pairs, which means it is 10,000 base pairs in length. There will be a region which will be called as a promoter. And this will be, again, remember, this whole stretch is nothing but ATGCs. It's just ATGCs in different combinations, C, 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 T, T, T. So this is just DNA, okay? And in this DNA, we are basically trying to say that there will be defined regions which we can label with English, using the English language, and we'll call the region on the left hand side, and I'll define what the left hand side is in terms of orientation, as a promoter region. And after that, we will divide the rest of the gene into two basic categories. We'll call the ones we have labeled as blue as exons, and ones we have labeled in red as introns. Okay. And there will be another light blue region in the end, which is basically the terminator region. So this is pretty much the structure of a gene. A eukaryotic gene will have a promoter region, which will extend, the range varies, let's say 500 base pairs to 1 kilobase. So 1000 bases to 500 base pairs. We will have pieces which we'll call exons, and we'll have pieces which we'll call introns, and then we'll have a terminator region. Okay. And very obviously, the machine, DNA dependent RNA polymerase, is going to read this gene, and it's going to make a piece of RNA. The piece of RNA will have both these exons and introns. And again, this is a simplistic picture. And the net result is the immature pre-RNA, as we call it, will become the mature RNA after it removes the red pieces from the total information it is copied from the gene. So the mRNA, which is now going to be translated into a protein, basically is missing these red pieces, and which is why, looking back, knowing that these pieces are no longer represented in the code, we call these as introns. So basically, these are pieces of information which are available on the genome, on the DNA, but are not used to make a protein. So effectively, there is an editing step which we term as splicing, which happens, and this editing step removes so-called unwanted information from the pre-RNA to make the mature RNA, which is going to be converted into a protein. Okay. And again, these are all approximate sizes. Let's say the gene encompasses about 10 kilobases. The RNA will pretty much encompass something short of 10,000 bases. And after splicing, a lot of RNA would have been removed before it is converted into protein. And you end up with a much smaller piece of nucleic acid, single-stranded mRNA. So you start with double-stranded DNA. You use one of the strands to copy a complement, which is RNA. The complement is spliced, which continues to be the mature RNA. And the mature RNA is translated by the ribosome into a linear polypeptide chain, which is an amino acid chain, which then folds into a special structure called as a folded state of a protein. And this is all we are going to do in this class and in the next class. Is there a question? All right. So now let's look at more detailed terminology. And this may look complicated, but it's not really all that complicated. And hopefully you'll be able to, for those of you who haven't done biology before, you'll be able to follow it. Now, pretty much this is the region of interest, which is going to make the RNA. Fine. The location where the protein binds is approximately in this location. So there is a stretch of DNA which works as a binding site for DNA dependent RNA polymerase. Fine. Now, once RNA polymerase binds, it opens up the double stranded DNA and starts reading one of the strands. And it makes a copy, an mRNA copy of that one strand of DNA. And the first part of it is usually called the non-coding sequence, because this part never really is converted into protein. Then come the pink exons over here, sorry, the green exons over here, and the pink introns. All of them are copied, including the so-called non-coding sequence. And at the end, the RNA will contain the 5' region, the 3' region, the main gene, but the introns are going to be spliced out. Okay, I'll explain what enhancer regions are a little later. So this is the structure of a gene, which basically consists of introns and exons, 5' and 3' leader sequences, a piece of DNA which is there to bind RNA polymerase, and all this is nothing but a single stretch of double-stranded DNA, out of which only one strand is being read and copied into mRNA. So now let's look at a simple example of transcription.
Before outlining DNA transcription, I feel it's important to give an overview of protein synthesis to put it in context. First of all, we have the DNA, and where we find a gene on the DNA, this would be transcribed to form a piece of mRNA. This is known as transcription. That mRNA would then move from the nucleus, where transcription took place, into the cytoplasm, where it's going to be used to form a polypeptide chain in a process known as translation. Ultimately, that polypeptide chain would be folded to form a protein. In this video, we're just going to outline the first part, the transcription of DNA to form a piece of mRNA. Transcription begins when a gene is located on DNA and that particular part of the DNA is unwound or unzipped by an enzyme. Once that particular area has been unzipped, you then find DNA as two single strands, and one of those strands is going to be used as a template upon which to form a piece of mRNA.
So please notice which strand in which direction is being copied.
Piece of mRNA is formed by the enzyme RNA polymerase by adding complementary RNA nucleotides to the template strand of the DNA. Notice that only one strand of the DNA is used to do this and that the other strand is not used at all. Furthermore, the piece of mRNA formed is single-stranded. In this animation, I've shown you the section of DNA to be transcribed, the gene. But remember that DNA contains many genes, and therefore the piece of mRNA formed will ultimately be shorter than the complete length of the DNA. Finally, notice that the DNA is using thymine as one of its nitrogenous bases, and the mRNA uses uracil instead. Once this mRNA has been formed, it moves out of the nucleus where this transcription has taken place and into the cytoplasm, where it will be later used in translation to form a polypeptide chain.
So
it's been formed, it moves out of the nucleus where this transcription will be later used in translation.
So to highlight the changes again, the sugar is in RNA is nucleic acid polymer. The sugar is going to be ribose instead of deoxyribose. That's one change. And instead of T, we are using uracil, a nucleotide which contains uracil. But pretty much RNA is a cousin of DNA. It usually exists in single-stranded state, but it always associates with proteins and folds fairly well. So the word single-stranded is basically textbook language. And what you could clearly see is how a piece of RNA was made from DNA. And in your mind, you should compare and contrast the two processes: DNA replication done by DNA-dependent DNA polymerase, and RNA replication, which is done by DNA-dependent RNA polymerase. Okay, so let's now just look at the machine which is doing all this work. And this machine is RNA polymerase. Now, RNA polymerase, this is a schematic on the left, uh, pretty much like two hands cupped together. And you can see in this schematic, there are gaps in the protein. This is a protein enzyme. And you can see DNA actually going into this enzyme, being opened up in a small bubble, a bubble which is not as big as a transcription bubble. And you can see a piece of RNA which is coming out, which is being copied from one of the strands, which is complementary to one of the strands, which is being read. And as RNA polymerase keeps on moving forward, it will keep on moving forward on the DNA. You will get an mRNA strand, which is a faithful copy of DNA, and you get back the DNA helix. And this is pretty much what the crystal structure, atomic structure of DNA polymerase looks like. And shown over here are the different domains, the different pieces of this enzyme. So it's basically multiple pieces of protein put together, and this is something we'll discuss later. You can see it's sitting on DNA. On DNA, you can see this very small bubble that it opens up, and you can also see the copied RNA, which is called as a RNA transcript, and this whole process is called as transcription. Okay, now this is a sort of a picture of this entire process. In yellow is a schematic of RNA polymerase. This is DNA which is opened up. This is the DNA RNA hybrid region where you have extension of RNA bases in the 3' direction, which is copied from this strand. Now, terminology is calling this the template strand. And the other strand over here, as you can see, is called as the coding strand, and it will be of no surprise to you that this sequence over here of RNA is exactly equivalent to the sequence over here, because the sequence in brown, light brown over here is basically a complement of the template strand, and obviously the coding strand has the same sequence as the RNA strand, except for, of course, the fact that you have uracils instead of thymine. Okay, and you can see these free NTPs which are being used, which are present in a pool. DNA is unwound, polymerase moves towards the right hand side, and as the polymerase makes RNA and moves ahead, the DNA is rewound back to its double helical shape. So, are there any questions at this point?
Sir, like in the making of the RNA joins to this template strand. So the RNA polymerase also acts as a helicase to remove the hydrogen bonds between them.
Yes, so there are actually, I'm giving you a simplified view. There are many proteins which are functional over here. There are actually about somewhere between 20 to 100 proteins which are working over here. RNA polymerase is the machine doing this particular job, but it is assisted by a large number of proteins. So there is what is called as a transcriptional initiation complex over here. So different proteins are different doing jobs, including opening and closing of DNA. But yes, RNA polymerase is also involved in this process. So even though I'm telling you this as a one protein doing one job, the primary job of this protein, but do realize that there are a series of associated proteins which are also helping this whole process along. So transcription is not only about RNA polymerase. Transcription is about a group of proteins with RNA polymerase doing the key job in this process. Okay, all right. So let's look at this second movie.
Transcription is the first step in the expression of a gene. It is the formation of a specific RNA sequence from a specific DNA sequence.
So I want you to now focus on the fact that this is a stretch of DNA. There is an area defined as a promoter. There is an area defined as a termination site. So pretty much your gene is now between these two areas. Okay. And let's see how polymerase goes and makes a copy of DNA.
Transcription initiation occurs at a promoter, a region on the DNA where RNA polymerase binds. RNA polymerase attaches to the promoter and begins to unwind the DNA. At the initiation site, the polymerase begins reading the DNA template strand and building a complementary RNA strand from free nucleoside triphosphates. The RNA strand grows by the addition of these substrate molecules to its 3' end. This is the elongation phase of transcription. Note that the bases in the nucleotides of the RNA strand form pairs with the bases in the DNA strand. Also, note that the RNA nucleotides contain the sugar ribose, which has a 2' hydroxyl group, whereas the DNA nucleotides contain the sugar deoxyribose, which lacks this hydroxyl group. RNA polymerase functions similarly to DNA polymerase. It adds nucleotides to the 3' hydroxyl group of the last nucleotide. The DNA double helix rewinds as the RNA polymerase moves through. Like the unwinding, the rewinding is an energy-requiring process that is accomplished by RNA polymerase. When RNA polymerase reaches the termination site, the RNA transcript is released from the template. The DNA rewinds completely, and the RNA polymerase dissociates from it. The DNA and RNA polymerase can then participate in other rounds of transcription.
All right. So hopefully this was fairly straightforward and clear. It introduced you to the concept of promoter sites, termination sites. Let's now look at a more complicated movie.
What you are about to see is DNA's most extraordinary secret: how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
All right. Another movie slightly different from the first one.
The central dogma of molecular biology. The central dogma of molecular biology: DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated.
So in this part of the movie, the sort of, if this is a race, the person who's firing the gun appears to be coming from a slightly different side. You have already seen a large number of proteins, including transcription factors, the mediator complex, and of course, RNA polymerase, all assembling over here. But the way this is described is everything is ready, but the race cannot start unless it is triggered. And the trigger, the way they have shown it in this movie, is coming from a protein which is a little far away, which is actually bound to a stretch of DNA called as the enhancer DNA. Now you'll see this enhancer protein sitting on DNA, which is called as enhancer DNA, coming and triggering this whole process.
The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
Now remember that what the polymerase is doing is everything starts with the polymerase binding to the promoter. There is an initiation site, then copying starts, and there is a termination site where copying stops. Now, the size of genes vary dramatically, as I said, from a few hundred base pairs to tens and thousands to 100,000 base pairs. And the polymerase, whether it is a short gene or a long gene, its job is to stay on the DNA and keep on making RNA till it hits a sequence which tells it that it's a termination step in making RNA. So it's hit the termination piece of DNA at this point.